So I'm going to talk to you today about the Oxford AstraZeneca vaccine journey that I've been on for the last year and a half. But first, I'm going to take a little bit of a moment to talk to you about why I believe vaccines are one of the most effective medicinal interventions ever. This is a very old, very simple infographic from Public Health England showing the number of vaccine preventable diseases before a vaccine was introduced and then after a vaccine was introduced. And I think we've seen the same across the SARS-CoV-2 pandemic. We've seen the huge impact that vaccines can have in preventing hospitalizations and deaths. But when you're designing a vaccine, what are the characteristics that you need to consider? Well, first and foremost, it's safety and tolerability. There's no point in you designing a vaccine that causes side effects that are more severe than the disease you're trying to protect against, or will prevent individuals following up and having a full vaccination regime. Ideally, you want your vaccine to protect against severe disease, and for that protection to be sustained or long-lived. And to do that, you need to be able to induce an effective immune response. So you ideally want antibodies and T cells. And then these are the considerations that I certainly didn't have first and front in my mind when I started making vaccines over a decade ago. But the first thing you need to consider is the cost, because there is no point in developing a vaccine against a disease that occurs primarily in low and middle income countries if those individuals can't afford to get it. You need your vaccines to be stable, so you want them to have a long shelf life. And ideally, you don't want them to require a lot of cold chain storage, because that can impact the cost. It can push it up. You need to consider how your vaccine is going to be delivered. Are you going to give it intramuscularly? Are you going to give it up the nose? Because again, that all plays back into the cost of these vaccines. I do want to touch very briefly and very lightly on vaccine hesitancy or vaccine reluctance, and to say that this is nothing new. This is a cartoon or an infographic of Edward Jenner. And he's regularly referred to as the grandfather of vaccination. And Edward Jenner in the late 1700s, early 1800s, noticed that milkmaids, when they'd been exposed to cowpox, did not go on to develop smallpox. And smallpox is thought to have killed millions and millions of people across the world. But a cartoon back then depicting individuals who had received this vaccine suggests that they go on to develop bovine-like characteristics. This is absolutely untrue, and actually smallpox is one of the very few diseases that have been successfully eradicated from the world through vaccination. So my work in outbreak pathogens really started in earnest in around 2012, 2013. And 2013, 2014 was the year that the Ebola outbreak happened in Western Africa. And after the Ebola outbreak had been contained, the WHO and other government bodies came together and essentially listed out those pathogens, those viral pathogens that they thought might cause the next pandemic or epidemic. And I'm sure members of the audience will have heard of some of these viruses, things like Ebola virus, SARS-CoV-1, probably Zika, and then some other viruses in this list don't necessarily roll off your tongue. Crimean Congo hemorrhagic fever, and likely Rift Valley fever is lesser known. But really what we wanted to do within the university was to develop vaccines against all of these pathogens. And I've been involved in all the programs for all of these pathogens, save perhaps Zika. I don't think I've done a lot there. But the other thing that we wanted to do was to develop a vaccine platform technology. So in English, that means a vaccine platform that we could make against any disease, or we could use against any disease, because we and others were convinced that the disease X would happen. That was the unknown. We didn't know whether it would come from an animal. We didn't know where or when it would arise. And really, we needed a platform technology that we could rapidly adapt to make a vaccine against this disease so that we could scale it and get the vaccine out to those who needed it. So how do I make a vaccine? It's a relatively simple procedure. The first thing I need to know is I need to know a little bit about the virus or the pathogen that I'm trying to make the vaccine against. So I'm going to take coronavirus as the example here, not surprisingly. So what I need to know about this pathogen is, or what I need to have a best guess at, is what part of that pathogen, if an individual had a strong immune response against, would they likely be protected? And the part of the pathogen for all the coronavirus vaccines that I've been involved in that we've targeted is the spike protein. And the spike protein is this very small part of the virus on the outside that mediates its entry into your body. This is how the virus gets into your body, disseminates through your body, and causes disease. And if you can block that interaction, if you can stop the spike protein doing its job, you're likely to protect people from overt disease. So how do I use that information to make a vaccine? Well, the platform technology that I routinely use is an adenovirus. And essentially, I take the genetic code for this small part of SARS-CoV-2 and I stick it into, if you will, this Trojan horse. And this coding sequence, or this essentially recipe for the spike protein, effectively what happens is the adenovirus carries it into your body through the injection, and your body will then express this really small part of SARS-CoV-2. Your body will recognize it as foreign, raise an immune response against it, so then that's ready and waiting if you become exposed to SARS-CoV-2. So adenoviruses are obviously a platform they're my favorite platform, but they are not without some disadvantages. So you and I and everybody in the audience will likely have been exposed to human adenoviruses before. And that can result in a phenomenon called pre-existing immunity. So what that means is if we're to use human adenovirus as a platform technology, it can get cleared relatively quickly from your body. And it can mean that you don't get a strong immune response towards the cargo, towards the spike protein as you might want. So we took a different approach. And instead of using an adenovirus that was derived from humans, we used one that was derived from chimpanzees, so from monkeys. And the reason that I like this platform is you get high levels of transgene expression. Again, in English, that means you get a lot of spike protein being expressed with this platform technology. And in turn, that means you get really strong antibody and T-cell responses. And antibodies can be thought of as the first line of defense against infection. Essentially, antibodies, in particular neutralizing antibodies, will bind to the outside of the SARS-CoV-2, predominantly to the spike protein, and stop it getting into your cells, so stopping the virus in its tracks. T cells can be almost thought of as the second line of defense. So what they're really good at doing is recognizing those cells in your body that have become infected, and the T cells will attack them and clear them out of your body, so you don't have as much virus in your body and you get a lesser disease. You're not as ill. Adenoviruses also have a well-established reactogenicity profile. Again, in English, that means we generally know how much of a punch they're going to pack. You're going to get a sore arm, you're going to get a headache, and you're going to feel a bit miserable for a couple of days. The last reason is probably one of the most important. This platform technology is easy to manipulate and produce, and that means we can get lots of vaccine doses when we're scaling up this tech. So, for lots of reasons that I've just mentioned, adenoviruses are a platform of choice. And this was the development process and timeline that we were looking at pre-COVID, pre-SARS-CoV-2. Just to take you through this slightly step by step, you would invest time in an iterative vaccine design program, making sure that you really had nailed this before you moved on to preclinical studies. And for the preclinical studies, you would start small. You'd want to demonstrate in small animals that the immune response you were getting was of the type and the magnitude that you'd want. Once you demonstrate that, you would scale it up. You typically do studies in non-human primates, in monkeys. And you'd ideally want to do them in monkeys where you could then expose those animals to a challenge. So see what happened when they were exposed to the disease that you were trying to protect against. Only after you'd had favorable results at this stage would you go on to phase 1 GMP production. And that's the generation of vaccine doses that can go into humans. And the reason that you do it this way is because of the cost. We're not talking about incremental costs between each of these stages. We're talking about orders of magnitude. So you really need to be sure that you're getting the best results in the preceding steps before you go on to phase 3 GMP, because that's astronomically expensive. Prior to SARS-CoV-2, I'd worked on a vaccine against a different coronavirus. We'd used the platform technology, ChAdOx1, to make a vaccine against Middle Eastern Respiratory Syndrome, which is caused by a MERS coronavirus. And this is a lesser known coronavirus, but it is hugely impactful. It's got a case fatality rate of about 30%. The zoonotic reservoir, or where it's found in nature, are camels. And it's generally individuals that work with camels that have comorbidities that will succumb to this infection. So we do what we normally do. We identify which part of the MERS coronavirus we think, if you've got a strong immune response against, you'll be protected. And it's the spike protein that we used. We put it into our platform technology, our Trojan horse, if you will, and we started small. We started vaccinating mice. We then moved on to monkeys after we got favorable readouts here. And we've also vaccinated camels, the natural reservoir of this virus. And what did we see? We saw that we were getting a strong immune response, and in both of these animal models, they were protected against disease. This was the type of encouraging results that prompted us to apply for and get more funding to make a batch of vaccine that could go into humans. So we have a small research facility, we have a small facility called CBF that's localized on the, it's essentially 2 minutes outside of my office, that can make these batches of vaccine. We started small. We vaccinated about 24 people. The vaccine was as immunogenic as we thought it would be. So we were getting strong antibodies and T cells, and it was packing as much of a punch. So it was mild to moderate reactogenicity, sore arm, feeling a little bit unwell, generally cleared up pretty quickly. So we had these studies completed, or were in the process of running these samples when SARS-CoV-2 struck. And really, we almost needed to go back and reassess the development process and timeline. And we needed to think about how fast could we go? We did not take any shortcuts when it comes to safety or any of the regulatory requirements to push this vaccine through testing. The only risk we took was financial. And it wasn't my money. It was the UK government's money, AstraZeneca's, and also our partner Serum Institute. And what we did was, instead of doing each of these steps sequentially, we did them in this manner. So while we were running the preclinical studies, we were making our small phase 1 GMP, again, just across the road, at a cost of about £1.5 million. I was relatively confident that I could design a vaccine, and I was relatively confident that the vaccine would be immunogenic in mice, because that's what I do, and I have been doing it for years. It's what we all do within the team. Was I confident that we would have smooth sailing all the way through? No, and that's what we need the clinical trials for. So early in January, and I get in a little bit of trouble for this because I say that the information I needed to make the vaccine arrived into my inbox on a Friday night. But truth be told, I was up past midnight, so it was early Saturday morning that the information arrived. Regardless, the information arrived into my inbox late Friday, early Saturday morning. Saturday morning, I got up, started co-designing the vaccine over the weekend. I actually thought the job was done by late Saturday night. But on Sunday morning, more information came in, more data arrived, essentially more recipes for this spike protein arrived into my inbox, and I had to pick one. I had to pick one coding sequence that we were going to go with, and I did. And then by Monday, January 13th, we were ready to go. We were ready to order the reagents that we needed to make our vaccine. And at that stage, cases worldwide were low. we're at 41 cases. So over the next number of months, I worked with a number of different groups pushing through the preclinical studies. But essentially, the first study started on the 17th of February. The cases were at about 80,000 worldwide. And to say the team was small is an understatement at this stage. This was me making up the first vaccine to go into an animal model. I handed the vaccine to Marta. She went off and vaccinated mice. And that's how the story started. 9 days later, we were able to show that the vaccine was immunogenic. Over the next number of months, I worked with a number of collaborators and colleagues across the world, endless phone calls to Northern America, to Australia, and to colleagues within the UK to get this vaccine tested. We don't routinely do as much animal testing as I'm describing here, but at this stage, there was a theoretical concern of a phenomena called vaccine-enhanced disease, and that might be seen with SARS-CoV-2 vaccines. What is vaccine-enhanced disease? It can happen in some animals and humans when they get vaccinated against a pathogen and then are exposed to it. So when they see the virus or the pathogen, they get sicker than if they had not been vaccinated. And this had been shown to occur with a small number of animal models with experimental SARS-CoV-1 vaccines, largely inactivated vaccines, so a different platform. But really, we needed to look at the immune responses in animals to make sure we weren't seeing this type of pathology. And the immune response that was largely associated with this overt pathology was a Th2 type immune response. So we vaccinated monkeys. We have vaccinated monkeys, hamsters, mice, ferrets, and pigs. And what we were able to demonstrate was that most of the responses, all of the responses, were predominantly Th1. So a response that is not associated with vaccine-enhanced disease. But really what we needed to do was a challenge model. So we needed to vaccinate monkeys and to then expose them to SARS-CoV-2 and see if they were more poorly than unvaccinated controls. And you have to remember that at this stage, we're in late Feb, early March. The people that I was working with, the teams that were coming together, they normally have months, years to get these animal models together. And they were going at unprecedented speed so we and others could test these vaccines so that we could essentially develop a vaccine in case we needed it. And we didn't know then how far this virus was going to spread. So in late March, early April, cases were creeping up. We got the news from the Rocky Mountain labs of the monkeys that had been vaccinated and challenged. And what did we see? So first of all, from an immunologist point of view, I was very happy because we were able to demonstrate that we were getting good antibodies after one shot, but stronger antibodies after 2 shots, and they were neutralizing antibodies. But most importantly, we did not see overt pathology. We did not see vaccine-enhanced disease. And in fact, what we did see was that these monkeys were actually we prevented pneumonia caused by SARS-CoV-2. So they were protected from disease. So this data was really impactful for me. And it was at this stage in the journey that I remember that the media were very interested in these results. And this data tended to get reported with varying honesty around the globe. We soon went into a lockdown within the UK. And my memory of that lockdown is the streets were empty, which was both scary and good because it meant I didn't have to look as I crossed the road when I walked to work. Cases, as I've mentioned, were at 345,000 around the world at this stage. While we were doing the preclinical work, while we were pushing through all of these animal studies, Kath at CBF was making the first batch of vaccine. And that vaccine was released to trial. So this is vaccine that can go into humans on the 22nd of April. Some days later, an agreement was put in place to partner with AstraZeneca. And that, for me, that partnership has been phenomenal. Without the input and the drive of AstraZeneca, this vaccine wouldn't have got where we wanted it to get. We wanted it to make it available to low and middle income countries. We wanted to make sure that no profit was made during the pandemic, and that the vaccine was provided at cost to low and middle income countries in perpetuity after the pandemic. And AZ were wholly committed to doing this as well. Cases at this stage had crept up to over 2.5 million globally. On the 23rd of April, our first 2 volunteers got vaccinated. One got vaccinated with a placebo, so a vaccine that shouldn't protect you against disease. And another got vaccinated with our test vaccine, the ChAdOx1-nCoV-19. Within 24 to 48 hours later, there was false news reports suggesting that our volunteers had died. And this diverted a lot of effort of the team to disprove this and to reinstill confidence in our volunteers that were coming forward. I had my head down at this stage, and across the next 3 months, trying to get the assays and tests in place so that we could measure the immune response in these volunteers. And again, this scenario is similar to the animal models. You normally have months to years to set these types of assays and tests up. And there was endless calls with CROs, clinical research organizations, trying to assess if their tests were fit for purpose, if they were at the pre-specified level that we needed for our clinical trials. We were also setting them up in our lab as well. And we were doing this with Beth Kelly in AstraZeneca, and a lot of calls with Operation Warp Speed in the US. So not a lot of sleep as well. So the first studies that we were able to share with the world came out in early July. It's probably mid to late July, truth be told. Cases were quite high at this stage. And what did we demonstrate in this study? Well, what we looked at was the safety and the immunogenicity of our vaccine. It did what we thought it would do. It packed a bit of a punch with the first dose, but not as much of a punch with the second dose. And we also looked at the immune response. I do have to take a moment here to explain that our trials, and in fact all the trials, we define disease slightly differently. And the aim of our trial was to assess the safety, tolerability, reactogenicity of the candidate vaccine. So we're looking at all of these things of our vaccine. And the way that we were also obviously looking for efficacy. And the way that we defined disease was a PCR positive swab and one of the following. So fever, cough, shortness of breath, loss of taste or loss of smell. And that meant that we could pick up mild, moderate, and severe disease. And so when we quote our efficacy results, it's efficacy against mild, moderate, and severe. And for other trials, the way that they've defined disease, they generally, or in some trials, will pick up only moderate or severe disease. So when you, and I'm not saying you do, but if you compare efficacy across trials, you really need to get into the weeds and see if you're comparing like with like, because in a large number of cases, it's not feasible. These were the immune response that came out in that first study. It's a little bit of a dirty slide, and I do that to remind me about the work that we were doing to even set these assays up, to have them in place so we could look at the antibodies. We saw that after the first dose of vaccine, you were seeing a high level of binding antibodies and a large proportion of neutralizing antibodies. But at that stage, we only had a small group, 10 individuals who'd got 2 doses. And in fact, their antibodies were slightly higher, encouraging results to go for a 2-dose regime. We also looked at T cells, and we were able to show that we were inducing them. While we were doing these readouts from our phase 1 trial, our phase 2 trial had started back in June. And our phase 2/3 trial was a multi-country trial. We set it up with Suan Clemens, who is a force of nature in Brazil. She can vaccinate thousands of people in a day. We also worked with Shabir in South Africa, another fabulous collaborator. And across June, these trials really got set up in earnest. So while we were still testing and delivering the immune responses in our phase 1, we were also rolling the vaccine out into our phase 2 and our phase 3 testing. The next touch point for me actually came sometime after the initiation of the phase 2-3 work. For me, it was really important to demonstrate that this vaccine worked in older adults. There had been a lot of miscommunication and false news about how this vaccine worked in older adults, and we really needed to push that data out. We needed to demonstrate that the vaccine worked in older adults, and that was because age is a significant risk factor for both morbidity and mortality after infection. And for me, we needed vaccines to protect our most vulnerable, protect older adults. So what did we show? I've been a bit cheeky. I don't have a picture of an older adult getting vaccinated, so I've used Professor Sir Andrew Pollard here, who I figured is closer to the age group than I am. So we were able to demonstrate in older adults that, if you listen to me, it's probably easier than these slides. They saw, we were measured less reactogenicity. So these are adults aged 18 to 55, 56 to 69, and 69 to 70. And you can see that the reactogenicity, or the punch that this vaccine packs, is less in older adults. Antibody responses after the first dose were generally lower in the older adults, but after the second dose, they came up to the same level. We were getting neutralizing antibodies, and we also saw good levels of T-cell response. On the 31st of October, we entered our second lockdown. I don't remember much of that lockdown because for that portion of the year, I was working with the team, both in AstraZeneca and here at the university, to get the samples that we needed tested and shipped so that we could do the efficacy readout. And there was thousands upon thousands upon thousands of these samples that we needed to ship. They never seemed to stop. And not only did we have to ship them, we had to take sticky labels off the outside of the tubes, put the serum, essentially the fluid in your body that holds the antibodies, into different tubes, and it really never seemed to end. But in essence, we were doing that to enable the efficacy readout. And the efficacy readout is essentially, you want to count the cases in the ChAdOx1 vaccinated arm and the cases in your placebo or your control arm. And you want to see that there are less cases in your vaccinated arm, in your ChAdOx arm. So the lead statistician rang me on the 22nd of November, whatever the Saturday is before this date. The trial had been unblinded on the Saturday, and Merrin, who is exactly the type of person that you want doing this type of analysis, she's cool, calm, collected, and can explain really complicated maths very easily. She rang me on a Sunday morning and started bringing me through the PowerPoint of the efficacy analysis. And I stopped her on slide 2 and said, do we have efficacy? And we did. Absolutely delighted, tired, relieved, and then really confused and a bit grumpy because Merrin was telling me 3 numbers and not 1. And I didn't understand until she brought me through the efficacy in more detail, the efficacy readout. At this stage, the world was close to 60 million cases. And I do want to take you a little bit through those results because they were a little bit complicated. And I don't know that they were very easily explained at the time. So the efficacy readout that we presented, the interim efficacy readout that we described in November, essentially, if you just focus on the UK results, within the UK, we had 2 different groups of individuals. We had one group who got a half dose and then a full dose, and the efficacy associated with that regime was about 90%. In the second group within the UK, and these are individuals who got 2 full doses, the efficacy was about 60%. When you combined all the results, you got efficacy of about 70%. When the trial was unblinded on that Saturday, there was an onus on us to report these results on the Monday because the data could have an impact on AZ share price. At that stage, we hadn't had time to dig into the data and do the tests that were required to really explain why we were seeing these differences across these regimes. With a little bit of time, we were able to demonstrate that those individuals that had a half dose followed by a standard dose, there was a longer interval between the 2 doses. And that was associated with higher antibodies. Those individuals that had 2 standard doses, there was a shorter interval, and they were associated with lower antibodies. And this was the interim efficacy result. The primary efficacy result was 67% against mild, moderate, and severe disease. But most importantly for me, efficacy against death was 100%. So we prevented people dying. There's been a large trial conducted by AZ in the US, and again, efficacy is reading out at around a very similar number, even in a different country with different ethnicity. So for me, the journey of the Oxford AstraZeneca vaccine across 2020 can be split up in 2 thirds. The first one was the design of the vaccine, the pushing through the preclinical work that we needed to be in place to be really sure we weren't seeing this vaccine-enhanced disease. The clinical trials, initiation and rollout, and the testing of those results to look at the immune response after the initial vaccination in COV1 or the phase 1 clinical trial. And then the delivery of the interim efficacy readout at the late November. These efficacy results contributed to the MHRA giving us emergency use approval on the 30th of December. And I was doing what most people were doing at that stage for New Year's Eve. I was celebrating having a couple of glasses of Prosecco and a Zoom call with my family in Ireland. But actually there is no rest for the wicked. Emails came up in that night. I was back to work for a small group of us on New Year's Day to ensure that the vaccine rolled out happened. And one of the things that we were hoping for a little bit of a break, a little bit of respite, but one of the things that has consumed us, certainly towards the first half of 2021, has been variants of concern. And immune mutation, a virus mutation to evade the immune system is always going to happen. It happens with other viruses. It was going to happen with this coronavirus. And there are a large number of these variants of concern now. There's alpha, beta, gamma, delta, and mu. And I think it's largely accepted, regardless of the vaccine that is used, the neutralizing antibodies, those antibodies that stop the virus getting into your body, when you measure them against the original strain, which is called Victoria here, or the Wuhan strain, you have a relatively high level of those neutralizing antibodies. Well, when you then test to see if these antibodies can neutralize the different variants of concern, there's a reduction in that readout. So they're not as well able to neutralize other variants of concern. But most importantly for me, you're still seeing vaccine efficacy. So we were able to demonstrate within our trial efficacy against the alpha variant. And in early 2021, data started coming out from a number of studies. So from Public Health Scotland, from Bristol, from Public Health England, demonstrating that the vaccines that were being rolled out were protecting people. They were keeping them safe. And more recently, this is a very simple, but I think a very good infographic, demonstrating the close coupling of cases per 100,000 and deaths per 10 million within the UK, and how closely related they are, but how during the third wave they've been decoupled due to vaccination. There's more and more real-world data coming out, effectiveness data, demonstrating that these vaccines are still effective against hospitalization or death, against a number of different variants. They're not perfect, but the vaccine effectiveness is still really impressive. Within the UK, the vaccine rollout program has been estimated to prevent 230,000 hospitalizations, 120,000 deaths, and over 23 million infections. And they're big numbers. And I don't think I'm ever going to be involved in a vaccine development or delivery program that really has this impact or reach again. We with AZ have been very committed to supporting equitable access globally. AZ have pushed through the approvals and emergency authorizations in over 100 countries to date. They've released more than 1.3 billion doses for supply in over 170 countries. At the moment, this vaccine is being rolled out to low and middle income countries through COVAX. COVAX is essentially it's a collaboration between Gavi, WHO, and other bodies to supply vaccine to low and middle income countries. And the Oxford AstraZeneca vaccine is supplying a lot of those vaccine doses. What am I doing now? I am working on a number of different tests in the lab, trying to see what happens in individuals who get 3 doses of the AZ vaccine. And what we've shown is that they get high antibodies after the first dose, higher after second, and higher still after third, and that you get higher neutralizing antibodies after a third dose as well. The other work that the team has delivered across the last year and a half is summarized on this slide. We've demonstrated or tested efficacy of vaccination against different lineages, both preclinically and clinically. We've demonstrated that the type of immune response in humans is that Th1 immune response, not that Th2 response, which we wanted to avoid. We've demonstrated that the vaccine is immunogenic and tolerable in individuals living with HIV, and the immunogenicity, the level of antibodies and T cells, is comparable in individuals living with HIV to those living without HIV. We've defined correlates of protection, and more recently I've been involved in work looking at mixed regimes, looking at what happens to the immune response when you've got an adenovirus and a RNA vaccine. What's the work that's cooking at the moment? We're looking at the persistence of the immune response. We're doing studies in immunocompromised adults, and we're also doing some studies in humans. And with that, it just remains for me to thank everybody who's been involved in this program across the last 18 months. But the biggest thanks have to go to the clinical trial volunteers, not just for this trial, but for all the trials, because without them rolling up their sleeves, we wouldn't have these vaccines. Without them stepping forward, we wouldn't be getting out of this pandemic. There's obviously an awful lot of other teams. You can't deliver this in a silo. You need an awful lot of teamwork to deliver it. And I just want to say thank you to all of those teams and people who have enabled that. And with that, I'm very happy to take any questions that may have come up.
Now, Teresa, thank you very much for that wonderful account of excellent science, excellent medicine, and excellence in all we do, which is what today has been about here. And speaking of that, I have a wodge of questions here for you. And I think we have about 5 minutes. So bear with me. I've got my glasses on. So if you bear with me, Teresa, I'm going to read 1 or 2 of these.
Feel free to call me Tess, by the way. My mum calls me Teresa or the taxman, so I'm feeling a bit naughty.
Okay. Now, the first question, actually, very relevant indeed. Do you think the speed with which this vaccine was developed will change the course for future vaccines and also for companies working together?
I hope so. I do worry slightly that everything that we've sacrificed, and this is the global we, this is shopkeepers, frontline healthcare workers, bus drivers, everybody. We've sacrificed so much, and we've demonstrated what the best of us can do. And I worry that we'll revert back to our old ways, and we'll go back into silos and thinking myopically. I think there are lots of lessons, lots of positives that we've learned, and we've also probably learned about some things we need to get better. But really, I don't want my kids or your kids or anybody's kids to have to go through this again. So we need to be in a better position for when the next pandemic happens, and it will happen.
Okay, another question, Teresa, very relevant to yourself again. Where to next?
Yeah, that's a good question.
What vaccine, what condition is next on your sights once we sort this current situation?
So I've just had an email in, and somebody stopped me in the corridor. I've had a vaccine released to trial, which means the vaccine can start to be used in clinical trials for a multivalent, so targeting more than one Ebola virus. So I'm actively working at the moment on developing, because Ebola virus, it's not just one virus, there's a number of them. So, and we need to generate protection against all of them. And then the other one that I'm focusing on a lot is the one that doesn't roll off your tongue, the Crimean Congo hemorrhagic fever. So I'm afraid there's too many viruses out there that would keep you awake at night that need working on.
Okay, and let me throw in one about that dreaded thing in Australia, particularly, but elsewhere, dengue. I'm sure it's not possible really to get a vaccine for that. Any thoughts on that disease?
I don't work very much in dengue, so I'm afraid I'd be remiss. I think there's a lot of vaccine development going on in that field, though.
Okay, that's shot my trip to. Now, a slightly personal question, if I may, coming from scientists and hospital staff around the country, when you started, did you really believe you could do this?
I'm somebody who, so I'm going to be slightly facetious. Yeah, I can design a vaccine. I can get it to work in an animal. Did I think that we'd get where we got in 11 months? I don't think I could have dreamed that. And it was only through all the teams globally coming together. But I'm definitely someone who takes one day at a time, one task at a time. You get this off your to-do list, you move on to the next, and you just keep going and keep going and keep going, and hopefully you'll get there.
And in your opinion, if people have to mix a first and a second vaccine, say 2 different vaccines at different stages, do you think this will have any impact in your opinion?
So there are studies coming out, and some have been published and I'm involved in some of them, demonstrating that the immune response, there's no deleterious. In fact, it looks almost better for the immune response. The reactogenicity, it can be that there's a bit more of a punch with these, because if you think about it, the Oxford AstraZeneca vaccine, you get the sore arm in the first dose. With the RNA vaccines, it's the second dose. So it can be a little bit miserable, but no, nothing that I would report that I've seen that is bad.
Okay, now this question is a little bit about keeping up with coronavirus. The question is, how confident are you that we won't meet a variant that may elude vaccines?
So there's only going to be so much space that this virus can change or mutate into it until it can no longer bind. So the spike protein binds to this thing called ACE2 predominantly, and that's how it gets into your body and into your cells and makes you feel miserable and can kill you. If you change that protein too much, then that binding can't happen and it can't get in, and it's not SARS-CoV-2 anymore. So there's only so far it can mutate. I think we've seen with some of the variants, we've seen them mutate in slightly different ways, but really I've stopped to an extent. I've stopped looking at the number of infections, and I just focus on the hospitalization and deaths. And that hasn't been ramping up hugely, certainly here. And we're seeing a lot of transmission within the UK.
Okay, moving on to clinical trials, Teresa, and side effects. Is there any link between side effects, for example, and antibody response? And what is your experience generally of side effects post-vaccination?
So you're asking if the sorer your arm or the worse the headache you get, do you get stronger antibodies? I don't know that we've seen that relationship. I don't know that we've looked very hard in earnest. So the side effects that you'll feel a bit miserable after you get this vaccine for the first couple of days, and generally you'll feel better. You'll feel like you've got a common cold. You might want to take a paracetamol, you might want to Netflix and stay in bed, but generally you'll be back to normal within 2 days.
Okay, and what about booster vaccines? What's your opinion on that? And do you think there are any risks attached to that again in the side effect profile?
Yeah, that's a good question. So for booster vaccines, I think there is a case to be made if we look to low and middle income countries where healthcare workers are providing care for people who are dying of this disease and they haven't got one vaccine in their arm. And one vaccine in your arm will confer a degree of protection. So I think for me, those are the people that I'd like to get these vaccines to. There is an argument to be made, and I agree with this point, that certain populations, certain parts of the population might need a third dose. So individuals that are perhaps immunocompromised, or individuals that are older who are higher at risk and have a declining antibody. Do I think that we should be mass vaccinating everybody with a third dose? No. Do I think those doses would be better off in a low and middle income country? Yes.
Now, I understand there have been very minor tweaks in vaccines so far, but the question is, do you expect the makeup of vaccines in general to change, similar to how, for example, the flu vaccine is developed each year? That's the question.
So one of the advantages of the tech that I used was the speed at which you can go. So we made this vaccine pretty quickly. Between getting the sequence and having a vaccine that could go into animals, I think it was like 30 days, 35 days. That's not to say that there aren't other techs that can be used to make vaccines, so inactivated vaccines, protein and adjuvant vaccines, that might be of use for this pathogen in the future, or certainly protein and adjuvant vaccines are a good technology. They just generally can't go at speed that we and others can go. And that's what we needed. We needed a vaccine. Every day counted, to be honest, because people were dying.
Okay, Teresa, I think we're more or less got to the end of the questions. There's one or two more chatty, tweety things that have come through. I mean, first of all, obviously, to thank you for all your work on the vaccine. We're all very proud of you here in North Kildare, should we call it, or South Dublin, approximately. How can someone be so clever? So with that, again, to thank you very much. We hope to see you over here in South Dublin at some stage. And if you do have time, please do stay on.